Now? Oh, this is too loud. All right. Feel like a rock star now. So I'm going to tell you about some of the work that we did about the mechanisms of memory consolidation during sleep in rodents. And one thing I'm going to talk a lot about is neuronal sequences of activity. These are prevalent in many different cognitive functions. For instance, learning songs in birds, planning motor actions in mice, or decision-making in rats. There's many other examples. And the one I'm going to talk about is the hippocampus in rats and its involvement in spatial coding. But keep in mind that this is just a model of a more general process. And we know that the hippocampus doesn't do only space coding. It's involved in all sorts of memories, except that in rodents it's very convenient to test spatial memory. So this is what I'm going to tell you about. So if you record from the hippocampus in a freely moving animal, which is what we do, you will observe something like this. So here you have an animal that shuttles back and forth between two water reservoirs, and the 6 small images at the bottom represent the firing rates of 6 simultaneously recorded cells, pyramidal cells in the hippocampus, and the color indicates the amount of firing that the neuron has in different places of the environment. So this is an overhead view of the same maze that is represented here. Black means the animal never explored this part of the environment outside the maze. Blue is where the animal ran, but the neuron never fired when the animal was in those areas. And as the color moves towards red, this indicates that the firing rate of the neuron increases. So for instance, this neuron fires maximally in the lower left corner of the maze. This one, for instance, near the lower water reservoir, and so on and so forth. So each of these neurons has one preferred area where it fires, which is called its firing field, or place field, for instance. So, and these neurons are called place cells. They were discovered by John O'Keefe, who was awarded the Nobel Prize for this incredible discovery. And together, he proposed that these neurons form the brain basis of a cognitive map of the environment. So just so that you understand what I'm talking about, when I'm walking here in the environment, if I'm the rat, and you're recording from one of my place cells, this neuron might fire when I come next to Itzhak, for instance. But this is not an Itzhak cell. If you turn off the light, and I keep walking around and come back here, the same neuron will fire again. So it's not a sensory neuron. It's really about where I am in space. One interesting thing is that when the animal moves about in the environment, successive subpopulations of neurons will fire as the animal enters their place fields. So the first neuron will fire here, then I move away, this neuron turns silent, but another neuron will start firing. Well, actually another set of neurons. And if you record with one electrode here in the hippocampus, you will notice 2 kinds of signals that I'm going to tell you about. The first one is this rhythmic signal, which is like an EEG, electroencephalogram, but it's inside the brain, so we call it a local field potential. And this oscillates very regularly at approximately 7 Hertz, and this oscillation is known as the Theta Rhythm. And it's prevalent in the hippocampus when the animal explores the environment. The second thing that you'll notice is that individual neurons emit action potentials. So this is the way they communicate. They emit small bips, which are like a Morse code, if you wish. All these bips are the same, but probably their intensity or their timing is what matters. So here, the animal enters the red place field, and the red neuron, the red place cell here, fires as the animal is within the field, as I explained before. But this is a trajectory, so there's many different neurons, and in fact, here the animal goes through the red, yellow, and green place fields, and the same neurons, red, yellow, and green, fire in turn, and this forms a sequence of activation. If you're reading out from the brain as an experimenter, for instance, you will be able to know what the animal is doing in space, how it's walking. You can follow the animal just by looking at the activity of these cells. Now, during sleep, and in particular during slow-wave sleep, what is the hippocampus doing? These neurons can no longer respond to what the animal is doing in space. Now the animal is not moving, is completely disconnected from the outside world, yet there's, of course, a lot of activity in the brain during sleep, and especially in the hippocampus. what you see on the field is something like this. Theta is gone. Instead, you have this irregular activity at a high frequency, approximately 200 hertz. This oscillation is called a ripple. The neurons are also active. They emit endogenous, spontaneous action potentials. But this does not happen randomly. First important thing you'll notice is that these spikes occur preferentially during ripples. So this is a period of time when the network activates spontaneously. It's no longer driven by any sensory information inputs. Even during wake, we can argue it's not driven by sensory inputs, but here it's even stronger. I mean, it's really the internal dynamics of the system. Not only this, but neurons don't fire randomly. If you look at what they're doing here, red, yellow, green, they actually recapitulate what is happening during wake, or what has happened just before during exploration. So it's as if the animals were dreaming. I borrow some quotes from you so that I don't get in trouble for saying this. I don't know if the rats do dream, but it's an image. It's as if they were dreaming that they are exploring the maze. They're virtually, navigating in the previous environment. And this is known as replay. So the first thing I tell you about, the first experiment that I'll describe, is one in which we wondered what the role of these replay events during sleep could be. As we all know, there's evidence that sleep is very good for memory consolidation, and there was a hypothesis put forward by Yuri Buzaki in 1989 that this activity in the hippocampus during ripples was probably instrumental for memory consolidation. So we set out to test explicitly and causally this prediction. So this is in the general framework of memory consolidation, which is thought to involve a dialogue between the hippocampus and the cortex, where initial memory traces would be formed in the hippocampus and later, or progressively, transferred or exchanged, I'm trying to use soft words, through a dialogue with the cortex for long-term storage. This does not mean that the hippocampus is no longer useful for long-term memories or that the cortex is not involved in early stages. But there's a need for a dialogue between the hippocampus and cortex so that memories are consolidated. And this could happen during sleep. So I'm going to first focus on this part of consolidation, which is the role of the hippocampus. So we have a proposed mechanism, which is these replay events during sleep. They could be instrumental for memory consolidation. How do we test this? Conceptually, it's pretty simple. If ripples are instrumental for memory consolidation, then disrupting ripples should have induce an impairment in memory consolidation. So how do we test this? Well, we record from the hippocampus, and we have a system that online detects ripples in real time. And whenever a ripple occurs, we can stimulate the ventral hippocampal commissure, and this is expected to interrupt the ripple and associated replay. So it's a very timely and precise operation. Then we need a behavioral task where we can assess memory consolidation. So this is a classical spatial reference memory task, where the animal has to find 3 food rewards in an 8-arm radial maze. So the animal explores this maze. Of course, we don't tell the animal where the rewards are in the first place. So the animal just walks around, discovers randomly that, oh, here there's a reward. Maybe notices that on day 2, the reward is located at the same place. And also, I should mention, there's a limited amount of time to get the rewards. So after 1 minute, the animal is taken away. So the best he can do is try and get all the rewards without visiting those arms where there's nothing to find, because it's always the same. Every day, it's going to be the same arms that are going to be rewarded. So this is a reference memory task in the way that whatever is learned on day 1 is useful for day 2, 3, and so on. So there's an incremental process, and you have to consolidate this information, because if you lose it from day to day, you're not going to improve. It looks like a simple task, but it takes some time for the animals to learn this. And in the control condition, the animals, after the training, they can just go to sleep and do as many ripples as their brain wishes. Now we have another group where, following training, during sleep, and only for 1 hour, for practical reasons, I may explain later. We are going to detect all ripples online and stimulate the ventral hippocampal commissure to suppress ripples in a very timely fashion. The suppression here, the stimulation is 0.1 milliseconds, so it's a very brief single pulse stimulation. You can see an example ripple here, and this is one that has been interrupted. It's zoomed in here. You can see 1, 2 cycles, and then bam, we stimulate, and the ripple is gone. Okay, on this figure, you can't see a thing, and that's good. It means there's no ripple left. Now, this is not sufficient. We cannot just compare the two groups, because there would be a big caveat. Imagine you find a difference between these two groups. It's always possible that there's something else that is happening. For instance, our stimulation may recruit something else, not just destroy the ripple, but have other uncontrolled effect on whatever other brain region. So we need a second control here, where we also stimulate whenever we detect a ripple, but the stimulation is slightly delayed in time so that it doesn't interfere with the ripple. So in this group, there will be as many stimulations as in the test group, but this will not suppress ripple and associated replay. This is what you see here. Can we stop it? Yes. Oh, it works. Sorry. What you can see here is a ripple, and then we detect it, and the stimulation happens 150 milliseconds later after the ripple is over. So it doesn't interfere. And you can see here in this figure that there's a lot of ripples occurring approximately 100 milliseconds or so before the stimulation. So this is effective. We detect the ripples and stimulate a bit later. Now, what happens with performance? So these animals are tested daily. And as you can see, their performance increases slowly over the course of 10, 12 days until they reach a plateau, almost perfect performance. This is the control group, the unimplanted animals. If we now look at the performance of the animals which are the controlled stimulated animals, stimulation does not induce any difference, any effect on behavior. However, I wouldn't be here otherwise. In the test group, you can see there's a detrimental effect. The animals, really, their performance decreases significantly compared to the other two groups. Now, of course, you might argue they do, but still, they do learn something. Sorry, this is wrong button. Where are we now? Oh, we're back. All right. So they do learn something. You know, they're out of the random area where animals are doing just random behavior. They did learn something. They improved a bit. There may be several reasons for that. One is, as I said, we stimulate only for 1 hour. So we don't suppress ripples and replay for 24 hours, if only because the hippocampus, as you know, is a zone that is very susceptible to seizures. And if we overstimulate, we're afraid that this might trigger seizures in those animals. So we don't want to do that. Also, we know that most ripples with replay occur during the first hour. There's only one study on this, but at least that's what we know. Another issue is that we can't destroy the entire ripple. As I said before, we first need to detect it. So there's at least a couple, 3 cycles that happen before we can actually interrupt it. So some information may have been conveyed. More importantly, maybe other brain structures take over. It may not be only the hippocampus that is involved here, and if we start interfering with the hippocampus, there may be other mechanisms. This could sound like hand-waving. However, there was a study published in 1995 where Jarrah did a complete lesion of the hippocampus and compared here 2 groups. Well, I'm going to flip this so you can compare more easily. And you can see this is the control group, and the test group is a group where the hippocampus was completely lesioned. So our stimulations are as effective as removing the entire hippocampus from these animals. So it's a very strong effect, actually. And this was the first evidence that ripples and associated replay are instrumental for memory consolidation. Okay, so ripples are one of the mechanisms of memory consolidation during sleep. I'm going to skip this. Let's go back to the idea that there's a dialogue between the hippocampus and the cortex. We haven't provided any evidence that the cortex is involved here. I think we have good evidence that the hippocampus plays a critical role, but what is the cortex doing? Is it really listening to what the hippocampus is saying, or maybe this is only intrahippocampal? Who knows? So in a second experiment, we set out to examine what was happening in the cortex and how this dialogue could take place between those two structures. This is a very long-standing hypothesis, but we wanted to get some evidence for this, direct evidence. So the theory is that during behavior, information is going to be encoded in the hippocampus and cortex. All these networks are very active. They start to form connections. And then during sleep, the hippocampus, where is the other one? The light no longer works. Okay, you'll have to imagine. So at the bottom here, you see the hippocampus reactivates. I can try to use the mouse instead. Okay. And this will in turn activate subnetworks in the cortex, which will update their functional connectivities. And this should be reflected the next day during behavior in the activity within the cortical network. So there's a reorganization, supposedly, of the subcortical networks, which will be useful for memory consolidation. There's evidence, correlative evidence for this scenario. One example here is in this task, so animals had to choose between two possible paths in a Y maze, and only one was rewarded, and there could be two different rules. In the first rule, the animal just had to follow, let's see this one first, had to follow the light. There's a light that turns on randomly on the right or left side. The animal has to follow the light, very easy. Well, so it seems. And the other rule is, well, you have to go right. No matter what the light is doing, just ignore the light and go right. And whenever the animal learned one rule, there was a switch to the other one. So switching and adapting requires the prefrontal cortex. You need some cognitive flexibility. Also, the prefrontal cortex is one of the major targets of hippocampal outputs, and there's very good evidence that it's involved in memory consolidation. I'm not saying here that the prefrontal cortex is the region for memory consolidation. I'm just saying that this is one of the best possible targets, so we studied the prefrontal cortex, but in no way am I saying that this is the only place where this is happening. It's an easier target. So during sleep, what was observed here is that whenever you see a ripple in the hippocampus, there's subsequent reactivation of groups of neurons in the cortex, in the prefrontal cortex. This is cell assembly. And if you look at what this cell assembly is doing during behavior, actually this is an assembly that fires whenever the animal reaches the decision point. And this is reactivated precisely after the hippocampus does its ripples. So this is consistent with the idea that the cortex is doing something relevant at the time where the hippocampus talks to it. Moreover, this only happens when the animal starts to understand the task. When it's doing random behavior, you don't see that. But this is correlative evidence, and we wanted causal evidence. Slowly, it's breaking apart. No, we no longer have. Okay, let me go on the other side. I think we have to change the battery. So again, the idea is that during learning, the hippocampus and prefrontal cortex will start accumulating memory traces and forming new networks. During sleep, there should be a dialogue between the two. And we already know that probably one of the components of this dialogue is the hippocampal ripple. And we have good candidates in the cortex as well. There are delta waves and spindles. These are cortical and thalamocortical rhythms that have been implied also in memory consolidation. There's some evidence that these rhythms are boosted during memory consolidation. So this could be one of the cortical responses to hippocampal ripples, even more so that there's a temporal correlation between them, although it's quite weak, but it exists. So our reasoning was this: since this coupling is weak, actually we could even improve this, enhance the coupling, and see how this affects learning and memory. First thing is we had to find a task where the animals would learn something but forget. So learning but no consolidation, and see if we can then induce consolidation by coupling hippocampal and cortical rhythms. We found such a task, which was developed by a group in Argentina. So it's a slight different version, variant on a classical memory task. So the classical version is that the animal is exploring an arena. There are two objects represented here in two adjacent corners. These are identical objects, and the animal spends 10, 20 minutes in the arena exploring the objects. Then on the next day, the animal goes back to the same environment, but now one of the objects has been displaced to a different corner. Typically, these animals are curious. What they do is spend most of their time around the displaced object. And this is good evidence that they remember where the objects were on the previous day, because they don't go to the object that is stable, that hasn't been moved. Now, the small change in this protocol is that if you train the animals only for 3-4 minutes, then this doesn't happen. The difference is the following. If you test the animals 1 hour later, they go to the displaced object. All right? So they have learned something. But if you test them the next day, they have forgotten. They visit both objects the same. So we thought this was a good task. We had evidence that the animals learned something but did not consolidate. It was forgotten. We said, that's the task we can use. First thing is we first verified the data. As you can see here, if they are trained for only 3 minutes, they don't discriminate between the two objects. It's 50% chance of visiting either object. Whereas if you train them for long enough, then they visit preferentially the displaced object. What happens now with hippocampocortical coupling between the rhythms that I mentioned before, ripples and delta spindles? Well, there's a huge increase in the number, in the incidence of coupling in the condition when there's memory consolidation. So we're on the right track, but this is still correlative. We haven't done anything causal here. It's like a first step. So the causal step now is, since we can do online triggering based stimulation based on ripple detection, now we're not going to destroy ripples. Instead, we're going to stimulate the motor cortex. I can explain later why motor cortex. And this will induce a delta wave and a spindle. This has been published long ago. And this pattern propagates. It's a traveling wave, so it will propagate to other cortical areas, including the prefrontal cortex. All we have to do is find the right timing so that the delta wave occurs in the prefrontal cortex at the right timing, same as endogenous coupling. And now we have a way to increase, boost this coupling. So whenever the coupling doesn't happen, we just stimulate at the right timing, and the cortex responds. Two conditions. Control condition is we stimulate at the right timing, and the test condition. The control condition, we also stimulate, again, for the same reason as before. You need to have stimulations, because what we want is not to say that the delta waves or the spindles are important. We want to say that it's actually the coupling, not the number of events, but the coupling within hippocampus and cortex. So we're going to have the same number of events in both groups, except in one group they're decoupled. In the other one, they have a very precise timing. So first prediction of the model is that during sleep, there should be a reorganization of cortical subnetworks. I'm going only to show a small part of this, just to give you a flavor for what we did, because it's slightly complex. But there's a measure that was defined by some of our colleagues, which is look at what happens during so the delta wave is this upward going deflection, and this corresponds to a period of silence in the network. It's a down state where cortical neurons are silent. Maybe I should recall this. During slow wave sleep, cortical neurons alternate between a down state where everybody is silent, or almost, it's hyperpolarized, and then there's an up state where everybody's active and starts talking again. And this alternates, and this is what actually constitutes the slow wave in slow wave oscillation. Right? So the delta wave is the silent component, the time where the network shuts off. But then activity comes back, and there's a spindle. So we're looking at this activity when the network turns back on. And this activity here is known to be very indicative of subnetwork functional reorganization. I'm not going to delve into the details, but we've reused this measure that was designed by others. And so we're going to compare this activity when we stimulate versus when we don't stimulate. Because there's also some endogenous activity. Sometimes there's a delta wave that occurs without our stimulation, right? So we're going to compare those that we induce versus those that occur spontaneously during the same time. So let's compare them. What we see now is that when we stimulate randomly, without taking into account what the hippocampus is doing, so now it's decoupled, there is mostly no difference between the activity in those delta waves that we stimulated, that we induced, and the endogenous delta waves that appeared randomly. See, so we have a very high similarity index, whatever that means. See, there's a very big peak at 1. It means it's all very similar. However, when we stimulate at the right timing, when we couple hippocampal and cortical activities, what we see is a bimodal population here. There's a large number of neurons that are not affected, which is expected. We don't think that every neuron will change its activity. But there is a significant subpopulation here that changes its activity tremendously when we stimulate. That is, when the delta wave is coupled to the hippocampal activity. You can see this here. So these are individual neurons, and the color indicates their firing rates. So this is the down state. Here they're mostly inactive. And then they resume their activity, and this is when the line turns red. Each neuron starts firing at a different time. And you can see here between the endogenous delta waves and those that we induced randomly, we don't see much difference. However, when it's coupled to the hippocampal ripple, here it's reorganized, it's different. In particular, the order in which they fire has changed. So we have evidence that there is some functional reorganization within the cortical network. This also is expected to be reflected in activity during behavior. So how did we measure this? We split the environment into 4 quadrants, and we focused on those quadrants where there was one of the objects, for instance, the displaced object versus neutral quadrants. Without explaining what these curves mean, I'm going to give you the message. All these curves look the same except one. There's one curve where the activity of the neurons are different, and this is only when we look at the displaced object after we've coupled the stimulations between hippocampus and cortex. So whatever we do, it doesn't matter. There's no difference except if we stimulate in a way that we couple the hippocampus and cortex. In that case only, you can see more activity of the prefrontal neurons around the displaced object. So now they respond to the task. What happens with behavior? Well, with behavior, in the control group, unstimulated animals, as I said before, they don't make the difference between the two objects. They forgot. Same thing for the group where we stimulated randomly. However, when we stimulate at the right timing, when we couple hippocampal and cortical activity rhythms, now the animals remember. This looks like magic. It did to us as well. We knew the theory, we had the idea, we said, okay, let's test this, and we started the experiment, and when it worked, we were stunned. I mean, it was just crazy. That on the next day, you stimulate a little bit during sleep, during 1 hour, and then the next day you see the animals, they go to the displaced object, they remember this. And actually, it's the same animals in both groups, except we randomly split them, we test them twice. Sometimes we stimulate at the right timing, sometimes not, and you can see they remember or they forget. Every single animal did that. So this is the first direct evidence that there is actually a dialogue between the hippocampus and the cortex during sleep that is instrumental for memory consolidation. This involves the hippocampal ripples with reactivation, and then the downstate and spinal in the cortex. How much time do I have? Do I have time to speak about something else, or is it? No, it's okay. You have 10 minutes. All right. Okay, I'm sorry, I lied in the beginning. When I presented the sequences during behavior, this was an oversimplification. So we have two kinds of sequences, I said. During behavior, the neurons fire one after the other when the rat walks across the room and enters different place fields, right? And this takes place at the behavioral timescale. It's constrained by the speed of the animal in space. Then during sleep, I said you have these replay events, which are very fast. Actually, this lasts a few dozen milliseconds, 30, 50 milliseconds or so. So it's accelerated at least 20 times. So it's really a fast-forward movie of what happened during wake behavior. But actually, this story is more complex than this. If you look at what happens in real life, the firing fields that I drew here don't look like this. They are not a beautiful tiling system where each neuron fires for a completely different area. They do overlap quite a bit. To an engineer, this might look like noise. You know, we're used to designing things right, and nature is noisy. Maybe not. Actually, one consequence of this is that the spike trains overlap in time quite a bit. So it's not one neuron that fires, then the next one, then the third one. They fire, but sometimes they fire together. And let's see what happens. Let's zoom in on this and play the movie slowly. The animal enters the red place field. Now the red neuron fires its first action potential. Then the animal moves forward a little bit. We're still within the red firing field, and the neuron keeps firing. But now the rat is also entering the yellow field, so the yellow neuron starts firing also. So both neurons are now firing together. A little further down the road, the animal enters the green field. It's still within the other two fields, so now the three neurons fire together, and so on and so forth. Now you can see here, this is not random. There is already some sequential organization here, and this is not only in my drawing, it's in the real data as well. You can observe this. Let's make it even more interesting. I said in the beginning that during behavior, there's a prominent rhythm in the hippocampus, which is the theta rhythm. So there's this oscillation at 8 hertz. If you factor theta in, you notice that these sequences are paced by the theta rhythm. There's actually one sequence per cycle. So they're called theta sequences. One thing that is stunning about these sequences is that if you look at what the neurons are doing within one theta cycle, the first neuron that fires here is the red neuron. And the red neuron, you may notice, its field is mostly behind the animal, so this is the animal's past. The next neuron that fires is the yellow neuron. This is where the animal is located now. It's right in the center of that field. So this is the present. The last neuron that fires is the green one, and its field is mostly ahead of the animal, so this is the future. So within a single cycle, you've got past, present, and future. We're still far away from a real algorithm, but we're getting closer, at least, to understanding why the hippocampus is involved in episodic memory. Of course, space alone is already a big component of episodic memory, but it's not sufficient. Here, we already incorporate time. So there's the past, the present, and the future, and these are very important components of episodic memory in humans. So let's go back to the previous picture, and now you see it's a little more complex. So we have two kinds of sequences at once during behavior. I'll let you consolidate a little bit because this is a lot of information. All right, so let's recapitulate. During behavior, we have two kinds of sequences. The first one is the behavioral timescale sequence. So as I'm moving around, one neuron will fire mostly one after the other. Although there's some overlap, mostly the neurons still fire one after the other, on average. At the same time, we have this theta organization, which is that at a fine time scale, 120 milliseconds or so, you've got the same sequence that is played constantly, very fast. Maybe I should emphasize that this is not trivial at all. I'm going to use my hands, but then I have to put the microphone down. So I'll try to shout. Imagine these are two place cells, two place fields. I'm saying that one is mostly before the other one, so there's something like this, right? And my fingers are individual spikes. The fact that one hand is to the right and the other one to the left doesn't mean that my fingers have to have the same organization. I can put my fingers however I want here. But I can also put them in a way that my left hand is always, my left fingers are always to the left of my right fingers. Or I can scramble them. So the order of my hands does not determine in a trivial way the order of my fingers. And that's what we're talking about when we talk about these theta sequences. The fact that one neuron, on average, fires before the other one doesn't mean that it's going to fire at a fine time scale before the other one. So it's a completely non-trivial phenomenon. Now the question is, we've got all these kinds of sequences. How do you replay a sequence during sleep? All right, I said there's sequence replayed during sleep, but how is it replayed? You have to memorize it in some way. You have to carry over what happened in awake behavior, keep it in your brain for a while until you go to sleep. You're no longer navigating. How is it memorized? So you need some initial formation of memory traces. What is the dynamical mechanism, network mechanism for this? We have two candidates here. The first one is the behavioral timescale sequence. And the other one is the theta timescale sequence. Maybe the most familiar one would be the one involving theta sequences. Why? Well, one thing that is great about these sequences, because they fire so fast, those neurons fire 20 milliseconds, 30 milliseconds apart. Normally, if it were only behavioral timescale sequences, neurons would fire several hundred milliseconds apart, or seconds apart, one after the other. How do you memorize activity when neurons fire at such temporal distances? The plasticity mechanisms that have been documented since the 70s involve very brief intervals between neurons when they fire one after the other to reinforce their connections. So maybe theta sequences are good for this, because now they allow the neurons to fire in very close temporal proximity and possibly form initial memory traces. On the other hand, 2 years ago, the McGee group showed a novel kind of plasticity. Now they claimed neurons can create long-standing connections, even if they fire seconds apart. And they call this behavioral timescale plasticity. It's a complex process that involves plateau potentials. But anyway, they've documented this kind of plasticity. So here, two mechanisms are actually possible. The neurons can form connections and remember the path, either through behavioral timescale plasticity or theta sequences. And our question was, which of this mechanism is the one that is instrumental to forming memory traces? To test this again, using a causal approach, but a funny one here, you'll see, the idea would be to destroy one of the sequences, for instance, the theta sequence, perturb that, but leave the behavioral timescale sequences intact. And see what happens with replay. You still get replay if you selectively remove theta sequences. But how do you remove theta sequences? That's a completely non-trivial question. And the answer is non-trivial and funny. You can see here, we're going to transport animal passively on a model train. And I'm going to show you what it looks like. Looks easy, but it takes 1 month to train this animal to learn this. It's absolutely non-trivial for a rat. What we knew from previous experiments we had done and that had failed, actually, is that when the animal is transported passively, the timing of the new spikes is perturbed. Whereas when he runs actively on this treadmill that we had to build, then the timing of the spikes is normal, as if the animal was just exploring the environment. So we said, okay, if we can perturb this fine timescale timing of these neurons, maybe this would affect theta sequences. So we have this protocol. The animal will either be transported passively or actively. By this, I mean when we turn on the treadmill. So by turning on or off the treadmill, we're hoping to selectively disrupt or allow theta sequences to occur. How do we see theta sequences? Okay, this is a bit of methodological information. So here we have several place fields, and the animal is now located here. We can record the activity within the hippocampal network, and you can see here the blue and orange neurons fire because the animal is located in this area. Now the animal moves a bit further down the alley here, and another set of neurons will start firing, and so on and so forth. The task the brain has to do, and the one we're going to do also, is do it the other way around. Now you know what the activity in the network is, and you try to guess where the animal is located in space. So here, my guess is that since the blue and orange neurons are firing, the animal should be somewhere around here. Wait, I also have a green spike there, so it's difficult. Where can the animal be? Maybe a bit to the right? I'm not so sure. So actually what I'm going to do is split this environment into little bins and assign a probability to each of these bins, that the animal is located here. So blue means I know, well, no, there's a 0 probability. The animal is certainly not located here. But then it might be around there. So each bin has its own probability. I'm turning this around, and now I'm showing you what happens during theta cycles. During theta cycles, if you take the activity of the neurons and decode, the way I just did, what you observe is the following thing. All right, I'll plot it twice so you see it better. You can see there's a tilt here. Why is that? Well, this is the beginning of the cycle. Where does the network estimate that the rat is located now? This dotted line is the real position of the animal. So the network thinks the animal is slightly behind its real position. Then, as we come to the center of the theta cycle, the estimation is much better. Now the network really reflects the current position of the animal. But near the end of the cycle, again, there's some kind of an error. The network estimates that the rat is located slightly ahead, and not where it is actually located. This should remind you something. It's behind, here, ahead. So it's the past, present, future. So this, again, is another way to show theta sequences. This is what the network is representing. It's representing the trajectory, the ongoing trajectory. I'm showing you this because this is how we then quantify theta sequences. So our goal was to selectively suppress theta sequences during this treadmill, well, when the treadmill was off, passive transportation. So here you can see when the treadmill is on, and you recognize these patterns which represent theta sequences. However, during passive transportation, you don't see that. Especially during the first session, there's nothing to be seen. It's complete noise. So the network no longer has these theta sequences. They're gone. I haven't shown that we still have the firing fields. They don't move. I forgot that slide. But you'll have to believe me. So the fields are not affected. So the behavioral timescale sequences are still there. The hippocampus at a very slow timescale still represents the position of the animal at its behavioral timescale. However, now we see that at theta timescale, it's gone. A little comment on these flowers. This is another way. I call that flower because it looks like it. Okay, this is another way to quantify theta sequences or sequences in general. What you should look for is red dots in this area. So this is where you have a nice slope. Remember, there was a slope in this colored figure. And also, it's linear. So whenever you have something that looks like this, a beautiful line going upward, then you'll see red dots up here. You don't see them during passive movement. Okay, so theta sequences are gone. What happens now with replay? That's the question. Does this affect replay, in which case we'll say that theta sequences are instrumental, or not, and then maybe theta sequences are a very beautiful epiphenomenon. These are replay events during sleep. These are significant replay events, so it's the same again, it's a virtual trajectory. They're not very beautiful, are they? This is after passive transportation, so when there was no theta sequence. Look at what happens after active transportation, when theta sequences were not suppressed. And again, after passive transportation. I'll show all of them together so you can form a better idea. These are the very few best examples we could find. There's actually very few of those. But there's plenty of these. So actually, only after, well, I'll ask you to trust me, and I'm not going to describe in details all these quantification figures, but only after active transportation do we see genuine replay, these sequences. Not only that, it's the only time when these sequences reflect the actual direction of motion of the animal in space. And also, this is the only time when these sequences look like the sequences that happened during behavior. Otherwise, you can see some sequences, but they don't reflect an actual trajectory in the environment. It's only some neurons that fire in a given order, but they don't correspond to what was going on during behavior. So the conclusion is that theta sequences during exploration are instrumental to form these initial memory traces that will be later replayed during sleep to enable memory consolidation through a dialogue with the cortex. Okay, I'm done. I thank you for your attention. I thank all the docs and postdocs who have made this beautiful work.